This is problem 3.9 from Horton's 5th edition by student request. Let me read the problem to you. Oxytocin is a non-peptide hormone involved in milk releasing response in lactating mammals. The sequence of a synthetic version of oxytocin is shown below. What is the net charge of this peptide at pH 2? So we're interested in pH 2.0. pH, that's A. B, pH 8.5. That's at 2. 8.5. And C, pH 10.7. Assume the ionizable groups have pKa values listed in table 3.2. So we should use table 3.2. I would say that those pKa values are those of free amino acids, and a more appropriate table in a polypeptide or a protein would be table 6.2. But the problem is rather difficult, 6.2, because in real proteins, amino acid side chain pKa's are quite variable. They have a range of values, so it's hard to come up with a precise answer. So they have you use 3.2, knowing that it's not really accurate, just to have a problem that can be solved. So there's some more information. The disulfide bond is stable at pH 2, 8.5, and 10.7. Notice that the C terminus is amidated. So it's done in 3-letter code. I'll read it out here in single-letter code. So remember, there's going to be an amino terminus. They say the C terminus is amidated. That's the other end. So the amino terminus is an ionizable group. It exists. And then there's a cysteine, a phenylalanine, an isoleucine, glutamate, asparagine, another cysteine, proline, histine, glycine, and they're reminding us that this is amidated. They say NH2, I'm going to put amide. And the disulfide, the two cysteines are involved in a disulfide linkage. So what do we care about? We care about the amino group because it's ionizable. So I'm going to write here, I'm going to make a table, amino. We care the phenylalanine and isoleucine not ionizable. The glutamate is, so we care about the glutamate. Asparagine is not, that's an amide. So it's carboxylic acid ionizable. It's an amide, it's not. The cysteines would be, let me get to that in a moment. The proline is not. The histidine is also ionizable. And the carboxy terminus is not. So we do not have to worry about thiols because the thiols, the cysteine, are involved in a disulfide bond. So they do not ion. There's no proton to ionize, and it's stable. So we don't have to worry about that. So the groups we have are an amino. So let me just draw it in this form. And this can either be in a protonated form, or it can be in an unprotonated form. And the charge of this will be either 0 or plus 1. Notice that I'm going to draw it like this because at a pH below its pKa, it will be positively charged. At a pH above its pKa, it's going to be neutral. The other group we care about is that of the glutamate side chain, and it can exist either in a ionized form or in the protonated form. And above its pKa then is a charge of -1, and below its pKa it's protonated and is neutral. So I'm going to say +1 here. And the other group we care about is that of histidine. Now this is a little more complicated, so I'm going to use a little more space for it. So I'm going to put that. Excuse me, I'm going to draw it in steps here so I don't get confused halfway through and draw the same form twice. So let me draw it like this. And we could have below its pKa, both nitrogens will be protonated. And I did something wrong here. Sorry, that's one bond there. Excuse me. This will be protonated, and above its pKa, only the it will be neutral. So it either has only these two forms. It's either positively charge a positive one or neutral above its pK. So let me translate that into here. So at pH 2, the amino group pH 2 will protonate the imidazole of the histidine. Oh, sorry, we're talking about the amino here. At pH 2, it will be in the protonated form, so the amino will contribute one positive charge. At pH 2, the glutamate will be neutral, and the histidine will be protonated, and the sum of those is going to be 2+. So this peptide, the synthetic peptide amidated disulfide bond at pH 2, it should have a charge of +2. At 8.5, the amino group, remember the amino, I didn't write down the pKa's here. I'm sorry, looking at table 3.2, we have the pKa, so let me draw them here. I forgot to tell you that, which you may be confused by what I was saying. So the pKa's of each of these groups, amino groups, according to table 3.2, is 10.7. The side chain of the glutamate is 4.1, and that of the histidine is 6.0. So the amino group is protonated at pH 2. The amino group is protonated. The glutamate is protonated, and the histidine is protonated. At 8.5, the amino group is still protonated. The glutamate is no longer protonated, so it is now negative, and histidine is no longer protonated, so it is neutral, and that means the molecule will have a net no charge. It will be neutral. At pH 10.7, we have a bit of a complication because that's the pKa. So let's do this a little more carefully. Let's do the glutamate first. The pH is now well above its pKa, so it has a negative charge. That for histidine, well above its pKa, so it is now unprotonated and neutral. And we have a little bit of confusion with amino. So at a pH equal to its pKa, we expect that half of it, so I'm going to write half of it will be still positively charged, and half of it will be neutral. And if we add that up, we will see that half of the molecules, then would be expected to be neutral, and half of them would be negative charge. The book says something a little bit odd here. It says the book says the answer should be negative 0.5. That can be misleading. That would imply that the molecule can have half a charge, which is not true in this case. What it means is that the average charge through time can be a half a negative charge, or any particular average of all molecules at once. So at any moment in time, half of them are neutral and half of them are negatively charged, which on average means they're this. That does not mean that any particular molecule or all molecules have half a negative charge.